Hello, my name is Richard Stone from Dana-Farber Cancer Institute in Boston. I'm pleased to be here at the first international workshop on acute leukemias. We've just had an interesting session about emerging therapies in acute myeloid leukemia. We know that there are 4 new drugs approved in 2017. We heard about several of them, plus one very interesting emerging drug, Venetoclax. I'm lucky enough to have Dr. Andrew Wei from Melbourne, Dr. Dr Levis from Baltimore, and Dr. Stephane de Botton from Paris to discuss some aspects of these emerging treatments. I'd like to ask Dr. Levis first, your impression of the FLT3 inhibitor story in AML. Where are we now with FLT3 inhibitors in AML in current practice?
Well, we're in a very busy place. We are encouraged by the fact that we have the first one across the finish line, midostaurin, but there's a swirl of other ones coming. I talked about the so-called big 4, midostaurin, quizartinib, crenolinib, and gilteritinib, but there are actually a few others along the way, but those are already in phase 3 studies. I think over the next few years, we're going to learn a lot about how to use these agents and when and in which patients with FLT3 mutations.
My second question here is, right now, which patients should be treated with a FLT3 inhibitor in the clinic today if they have a FLT3 mutation?
I think obviously any patient with the FLT3 ITD is going to benefit from midostaurin, and frankly a patient with a FLT3 TKD even more so. The question is, should that older patient who's got secondary AML and a FLT3 TKD, which isn't an uncommon situation, maybe they should really be getting Vyxeos. I think we would all be interested in seeing what Vyxeos plus midostaurin does. So it's still a bit confusing, but nonetheless, FLT3 mutations define a population of leukemia patients that you can really focus on. We're going to have a lot of treatment options, I think, coming soon.
So you have one mutationally targeted therapy, FLT3 inhibitor, midostaurin, to be used with chemotherapy in mutant FLT3 patients. We have another mutationally targeted therapy that was approved in 2017, Enasidenib, which Dr. de Botton has done a lot of work with. What patients should receive Enasidenib today?
The approval is for relapsed refractory patients. In the US and in Europe, it's more complicated beside clinical trials. It's clear that we should enter patients into clinical trials, and combination study will answer the question whether it's useful or not. Safety profile of Enasidenib is very interesting. The drug is not toxic at all, and the combo seems promising, as presented last ASH by Star. So they should be entered into clinical trial. That's very important frontline. And for relapse or refractory patients, there is an approval. So all these guys should receive that treatment. Whether they should be combined with something else, I can't answer yet. Health authorities in France are reluctant to do so. In Europe, generally speaking, in the US, I don't know.
So you showed some data looking at the characteristics that would predict response to Enasidenib and mutant IDH2 patients with relapsed refractory AML. So would you treat a relapsed refractory patient with AML who has either a high co-occurring mutation burden or a receptor tyrosine kinase mutation in addition to their IDH2 mutation with single agent Enasidenib?
Yes, I would, because we can never predict a response. Even if it's slower, you can never predict what will happen. So I would do so, yes.
Okay. Now there's a third drug that many people all over the world are excited about, Venetoclax, a BCL2 inhibitor, which you present some very elegant data, both preclinically and clinically using Venetoclax alone, but mainly in combination with other agents. Could you just take us through briefly, Dr. Wei, the preclinical rationale for using Venetoclax and the most important clinical data to date?
So most cells have a programmed mechanism of cell suicide, and this is prevented by a group of proteins called BCL2 family proteins, and there are at least 5 of those. And a new drug called Venetoclax has been developed which can specifically target one of those pro-survival proteins called BCL2. We know from phase 1 studies that on its own it produces modest response rates. However, in combination with other drugs such as standard therapies for AML, the hypothesis is that we can then target the remaining pro-survival proteins and release the effector BH3-only proteins, which can then go on to induce cell death.
So the clinical work with BCL2 in combination with cytotoxic agents, or at least some type of chemotherapy agents, what's the most important clinical data you want to talk about?
So the initial trials in combination have been done in elderly AML, which represents a major unmet need for the patient and physician community. And in combination with either hypomethylating agents or low-dose RSC, response rates compared to standard therapies have been potentially increased from between 11 to 19% with low-dose RSC, 28% with hypomethylating agents, to now 62 to 75% in combination with Venetoclax. The survival also looks promising, and the phase 3 trials are currently underway to demonstrate in a registration manner the potential for Venetoclax to hopefully change the practice for elderly AML.
Thank you. So I'm going to ask each of you one more question about clinical research in the field. First, Andrew, if a patient who's thought to be unfit for standard chemotherapy walks in the door today and you have access to Venetoclax, even though it's not approved in AML, it's approved in CLL in America at least, would you give them Venetoclax with a hypomethylating agent or low-dose RSC, or would you use the hypomethylating agent alone right now?
At the moment, we haven't got the full repertoire of information to help inform us. However, if the patient has intermediate risk karyotype, and particularly if they have a nuclear phosmin-1 mutation, I'd be fairly comfortable giving them low-dose RSC with Venetoclax because it's potentially cheaper and has advantages of being amenable to home delivery with the low-dose RSC. With patients with adverse karyotype, I think the situation is a little bit more difficult. At this stage, I would lean a little bit more towards a hypomethylating agent combination because of potential slight advantages there, at least from historical studies.
Dr. de Botton, there are many clinical trials that are going on now using IDH or IDH2 inhibitors in combination with other things. Of all that, which is the trial you're most interested in seeing the results of?
Probably in the elderly population, because the relapse rate is higher in that population.
So the elderly unfit population, you're saying hypomethylating agent plus or minus?
Even fit, because the relapse rate with chemo alone is very high. However, so this is the main focus I would be interested in, to demonstrate an advantage in with combination with IDH2 inhibitors or IDH1 inhibitors.
Thank you. And finally, Dr. Levis, there are lots of, as you pointed out today in your elegant talk, there's lots of ongoing clinical research with FLT3 inhibitors. Could you highlight one or two of the most important, either ongoing or soon to open trials, in your opinion, to answer the most important scientific questions with this patient population?
Well, I have a bias towards the post-transplant study because it will address 2 points. One, the value of MRD.
So that you say what the post-transplant study is.
The post-transplant study is where we're looking at gilteritinib versus placebo in patients who had an allotransplant with the endpoint of survival, but also with an endpoint of whether or not the presence of MRD predicts for relapse and also predicts for benefit from the inhibitor. So I think that's going to answer multiple questions, and it's one that really hasn't been addressed fairly yet. The second one is, I would say, quantum first, which is newly diagnosed patients where they get the FLT3 inhibitor all the way through versus placebo, which I think is what a lot of people want to treat. So I think I'm keen on those 2 particular trials.
Well, thank you very much. As you can see, we've had a lot of interesting data presented today and in the last year or two about new therapies in AML. Hopefully it's going to have an important clinical impact for our patients, and the early research, as you heard, is leading to a lot of subsequent research, which will be very important. Thank you.